Um, what does it take to move a rare disease treatment from an idea to an actual clinical trial? Welcome back to Rare Connection. I'm your host, Joanna, and this podcast is dedicated to connecting rare disease patients, educating both the public and medical professionals, and helping bring awareness to clinical trials for the 95% of rare conditions that don't have an FDA-approved treatment. Today, we're taking a deeper look into McCune-Albright syndrome, not just from a biological standpoint, but from the perspective of how treatments are actually developed. Joining us is Dr. Adebola Giwa, a board-certified pediatrician and rare disease consultant with 15 years of experience leading global clinical trials across endocrinology, musculoskeletal disorders, and other rare conditions. Dr. Giwa, for those who may be unfamiliar with McCune-Albright syndrome and what it makes it different from other conditions.
Okay, thank you. Hi, thanks, and thanks for welcoming to the show. Nice to talk to you and everybody, all your followers and listeners. It's a privilege to be here. So in regards to your question, McCune-Albright syndrome, yes, it's an ultra rare endocrine condition. So in regards to what it actually is and what happens, I think it's also important to think about what happens when you don't have McCune-Albright syndrome. So McCune-Albright syndrome is generally caused by a mutation in the GNAS gene. And what normally happens is you basically have, when you don't have it, you have a hormone, you can think of it, that goes and acts a receptor on a cell. That receptor then has some downstream effects, including the adenyl cyclase system, and it causes some end cell effect. And generally, once the hormone reacts with the receptor, it comes off and that whole process stops. However, with this mutation, what actually happens is you don't really have that initial stimulus from a hormone or other molecule, but that receptor is always activated and it's always causing, hey, create this downstream effect. So you can kind of think of it as the gas is always on without any of the breaks. So that can happen in a number of different cells in the body. And when that happens, it's called McCune-Albright syndrome when it happens in particular cells of the body. So with McCune-Albright syndrome, it's currently characterized by a triad of findings, which include effect in the bones, which is called fibrous dysplasia. It can happen in one bone or it can happen in several bones in the body. You can also see something called Café-au-lait spots, which are basically hyperpigmented macules or these big birthmarks that occur on your body. Or you can also see hyperfunction endocrinopathies, which basically means some endocrine hormones in your body are always secreting something. So in McCune-Albright syndrome, the diagnostic criteria is usually you have either one of the skeletal findings and one or more of the other things, or you can have the skin and the endocrinopathy findings without the skeletal findings. It's really a pretty broad um, phenotype you can see. So a lot of different manifestations of McCune-Albright syndrome can be found.
So we often hear the term mosaic mutation. What does that mean?
Okay, that's a good question. And mosaic conditions aren't seen very commonly, or when people think of genetic issues, it doesn't always pop up. So generally speaking, when a man and a woman come together to make a baby, the man brings his genetic material and a woman brings their genetic material. They come together, and then those two materials become one. And then that material, one becomes two, they divide. One becomes two, two becomes four, four becomes eight, etc. And if one of the parents has a genetic mutation, those one and one come together and that two become one and they multiply. And because they initially became one, generally speaking, all of the cells of the body will contain that mutation. However, when you have somebody who has a mosaic genetic condition, the process is different. So it's not genetically heritable. So generally speaking, the mom and dad do not have MAS. We'll use MAS as an example. So mom and dad come together with their genetic material. It does not have any mutations. These two become one, and then we'll say the one, then they multiply. One becomes two. The two become four. And now, after conception, after fertilization, then the mutation occurs. So now we'll say, just for this example, we have four pieces of genetic material, and number four has now the mutation there. So then they multiply, and the four become eight. However, the mutation is still only on four and number eight over here. So, and then those cells continue to multiply, et cetera, et cetera, et cetera. However, because the mutation happened after fertilization, all the cells do not have the mutation. It's only that particular lineage of cells that have the mutation and their further down offspring. So what you see is a variable number of presentations because of that. Because the mutation can happen at any time and any kind of grouping of cells can have it. So you can have affected cells and unaffected cells. You can kind of think of it as looking at mosaic tiles, actually. Some of the tiles have the pattern and some of the tiles do not have the pattern. So when you think of mosaic, the things that you should really hone in on is that it's not really genetically heritable, so it's not really mom or dad's fault or anything like that. And the mutation happens after fertilization. And because it happens after fertilization, it's the mutation, there'll be a mixture of cells that are normal and don't have the mutation, and a mixture of cells that do have the mutation. And because of that, the timing and where it occurs, the severity and the location of the mutations and subsequent effects can be very variable.
Hmm. So this I know is a de novo condition, meaning it's genetic, but it's not inherited.
That's correct.
So one of the more visible signs of McCune-Albright syndrome is on the skin. What is the characteristic findings people might notice or understand?
That's correct. So some of the notable findings include, just as you say, are on the skin. We call them cafe-au-lait spots. It's because of this kind of milky coffee looking color it can produce. And that's some of the notable things, depending on where the exposed skin actually is. You'll see it sometimes on the back, you can see it on the arm. You'll also see, because it's mosaic, there's certain affected cells. So it won't be all over the body. You'll usually see it patchy, and sometimes you'll see it respect the midline, and that sometimes it'll only be on one half of the body, which is interesting. Outside of that, the other characteristics of MAS also include involvement of the bone and endocrinopathies. So with so the other visible signs you could see also include when the bone is involved, so the skeletal manifestations. So you can see some significant skeletal deformities. Some are mild and some are a lot less mild in other people. So you can see that in a lot of different bones, actually. It can present in almost any of the bones of the body. You can see the cranial manifestation. So sometimes people may have, just as an example, really big one side of their jaw, or maybe a part of their head may look a bit more protrusive. You can also see scoliosis, so their spine may be a bit disformed. You can also see involvement of the legs, and you can see bone there as well. And then in regards to endocrinopathies, it's hard to really see it visually from the outside, but some endocrinopathies can make some of the other manifestations more visible. Particularly, you can have people can have issues with making too much growth hormone. If that's not well treated, when a person makes too much growth hormone, that can affect the development of their bones. So you'll see if they have involvement, for example, of their jaw, and they also have excessive growth hormone, that jaw can be even more protrusive and more notable.
Mhm. So with the cafe au lait spots, how do we tell the difference? How do you, what's the difference between them and something like vitiligo or a birthmark?
That's a good question. It all depends on context, I would have to say. So, for the example that you gave, you have the Café-au-lait spots, you have vitiligo, and you mentioned birthmarks as well. So, think about the Café-au-lait spots and the birthmarks. You can kind of think of them as uh, your melanocytes, the ones that make melanin, the cells that make your skin darker. Those are producing melanin that make those skin darker in both of those occasions. While vitiligo, depending on the reason why you have it, isn't could be an autoimmune condition, which basically destroys those cells. So you'll see that area actually have a whitening because they are not actually producing the melanin to make it brown. So with that being said, some people may have cafe au lait spots and no other symptoms. And that's not a concerning sign of some sort of pathology. There's lots of people that have cafe au lait spots and no other issues. Cafe au lait spots isn't something that necessarily needs to be treated, but it is a sign of a potentially larger issue if it presents with other things, such as the case in McCune-Albright syndrome.
So do those skin patterns tell us anything about how the condition develops at the cellular level?
It tells us a bit. Thinking about how I was using the example before that with the mosaic condition, you can think of it as tiles on a floor, and some are affected and some are not affected. When you look at the cafe au lait spots, you can tell that these cells were affected, and it was the cells that developed from those initial cells that were then also affected. So with that, you can kind of see the pattern of when you were just a fetus, where those cells may have been, and also how they may have migrated during that fetal period. So you can kind of see the pattern. And that kind of also explains why you'll see sometimes there'll be a respect of the midline, and some people who have it, they will not go to the other side of the body, for example, because it's just that lineage of cells that were affected. So it further shows a bit of how the mosaic uh, genetic spread happens through the body.
Mhm. So precocious puberty is often one of the first signs. What's actually causing the that in McCune-Albright syndrome?
Very good question. So for this one, I'd like to start with how uh, puberty normally happens, just at a really high level. So generally, your body and the endocrine system is very tightly regulated. I'm a pediatric endocrinologist, so this is the stuff that I love. Just figuring out what the hormones do and where they regulate different things. So generally, there's a part of your brain, your hypothalamus, that sends a signal to another part of your brain, the pituitary, that then sends two signals to your gonads. So in the woman, those are the ovaries. So once you reach a certain, and those signals then tell your ovaries, hey, ovaries, it's time that you become a woman. So we're going to need you to make woman hormones. So let's make a bunch of estrogen. So what happens in normal puberty is you're at a certain point, your body says, okay, you're ready to become a woman, and it releases those hormones, and then those hormones go to your ovaries, and your ovaries make estrogen. However, in McCune-Albright syndrome, your body, your brain is not telling the rest of your body, is not telling your ovaries, hey, this is the time that you go through puberty and become a woman. Your brain is not giving that signal. However, because of the mutation, the cells of the follicles of the ovaries, so in the ovary, you basically have these follicles, which include an oocyte, which is kind of like the genetic material of the person, and then supporting cells around it, which include, I'm going to geek out on you a little bit right now, the
Okay.
granulosa cells and the theca cells. And those cells work together to, one, produce androgens, or you can think testosterone, and then the other cell turns that testosterone into estrogen. So what happens in McCune-Albright syndrome is even though your brain is not releasing LH and FSH to tell those cells to make um, the androgens and to turn that androgen into estrogen, even though the brain is not telling you, those cells are doing it anyway. The gas pedal's on and saying, we're going to make this estrogen. So that's what happens in McCune-Albright syndrome. Those cells are activated and they're producing estrogen. And then because your body is producing estrogen, you're going to see signs of puberty. So in girls, you're going to see breast development. You're going to see growth spurt. And you're also going to see vaginal bleeding as well. That estrogen stimulates your uterus to grow. And then when because this estrogen production is cyclical, when that estrogen stops goes into a cyst and the cyst ruptures and the estrogen stops and there's a withdrawal, you'll have withdrawal bleeding. So basically what happens in MAS is you have inappropriate early estrogen production and signs of early puberty, which include breast development, growth spurts, and vaginal bleeding.
So how early can this begin, and what are the long-term concerns if it's not managed?
It can begin actually very early. Girls that develop, a lot of girls with MAS who develop peripheral precocious puberty will actually develop symptoms before the age of 3. So for those of you with kids out there, you can imagine what it's like, right now, maybe your kid or your kid was in that toddler age and you're working on toilet training and just cleaning and you're having issues with teaching them about vaginal bleeding and worrying about, do we need to get a training bra? So, which can be very stressful, as you might imagine. In regards to what happens if it's not properly treated, outside of dealing with all that anxiety of how do you tell their teachers how are you gonna have to clean up the vaginal bleeding with diaper changes? Is it can affect your growth long run in the long run. So what happens during puberty is well, in general, you have bones, and your bones are made of a mixture of chondrocyte, which is kind of like a spongy tissue, and then hard bone. So when you go through puberty, you produce estrogen. Estrogen causes those chondrocytes, that spongy tissue, to grow. And then also as it grows, it solidifies. That's how you get the hard bone. But eventually you're going to run out of those chondrocytes, which is why we don't grow to be 20 feet tall, at the end of puberty. So with that being said, once your bones are exposed to a certain amount of estrogen, it'll grow, but then it'll stop growing. So if you have puberty early, these girls will go through a growth spurt, but then they'll also stop growing early. So how that looks like from a practical standpoint is, say a girl develops precocious puberty at the age of 2, you know, now she's 5, 6, she's in first grade, second grade. She may be the tallest kid in her first, second grade, third grade class, but then she stops growing. So then by the time she's in junior high and high school, all of her friends then go through their growth spurt, but she has stopped growing. So what that means practically for her is she may be the tallest person in elementary school, but at the end she's going to have a shortened and adult height. So if it's not treated properly and they have that growth spurt early and they stop growing, their end height as an adult can be significantly compromised.
Mhm. So how tall do they normally get?
That's a good question. And it has to do a bit with a number of factors. One is what's your genetic potential? So how tall were you when you were first born? Um, so where are you, how tall are you supposed to be based off of how tall your parents are? Then also, when did the issues develop? So the longer time you have to grow, generally, the more time, the taller you will be. So what that means practically is somebody who develops precocious puberty at the age of 2 will have less of a height compared to somebody who may have developed precocious puberty at the age of 5. So, what you'll normally see is that these girls will generally be at the low range of their genetic potential. We calculate Pediatric endocrinologists do this all the time. We calculate the genetic potential by using a special calculator that takes mom's height, dad's height. We take into account their bone age, which is an X-ray of their wrists, which lets us know how old their bones actually look. So we use all that to see what your genetic potential actually is, and it's a range. So girls with McCune-Albright syndrome generally are at the low end of that range or below it. And what that looks practically is somewhere usually depending on a bunch of different factors, maybe somewhere uh, 5 feet, a little below 5 feet, a little above 5 feet. But
So how is this different from more typical forms of early puberty?
so when you think about early puberty, we generally separate it into two big categories. One is peripheral precocious puberty, which we were talking about, and the other is central precocious puberty, which we didn't really go into. So with the peripheral precocious puberty that we were talking about, the issue is that your ovaries are making estrogen by itself without direction from your brain, which makes it peripheral. When your brain is inappropriately telling you to go through puberty too early, the issue is central, which is why we call it central. The brain is the central mastermind of everything. Um,
Yeah.
so the most common type of precocious puberty, outside of MAS is central precocious puberty. So with that, your brain, for some reason or another, is telling your body to go through it differently. One of the bigger differences between peripheral and central precocious puberty are the treatments that are available to stop it. With peripheral precocious puberty, your body's making the estrogen, so most of the treatment is going to be directed at preventing the estrogen production or preventing estrogen at the location that it's supposed to act. While with central precocious puberty, you can use those treatments, but you can also use treatments that may try and stop the action at the central level. So there's different agonists we can do to try and disrupt that signal from being sent from your brain.
So we often hear about about this in girls. How does precocious puberty present in differently in boys?
So I'll speak specifically about MAS. So in both cases, you have somewhat similar cells that are overproducing or that are overactive. So first I'll start with the numbers and how those are different. So peripheral precocious puberty is one of the most common uh, findings in MAS in girls. Around 50 to 80% of girls with MAS will present with peripheral precocious puberty. However, in boys, it's some around 85% of them will have testicular involvement. But of that 85% that have testicular involvement, you may only see precocious puberty in a notable amount in 15% of the time. The other big difference is what cells are involved and what they actually produce. So in girls, the cells that are involved are um, the theca cells and the granulosa cells, and those cells are involved in making estrogen. However, in boys, it's different cells that are actually in the testicles that make androgens like testosterone. So they don't make estrogen. So that's one of the the big difference. Additionally, in girls, it's more episodic because you have the oocyte in the follicular unit that'll produce estrogen in this big burst and then kind of involute, and then another one will come eventually. However, in boys don't have oocytes or ovaries, they just have testicles. So it tends to be more chronic and a little bit more low level in regards to testosterone that's produced. So for the boys that have testicular involvement, what you'll see is macroorchidism, which basically means the testicles are enlarged. When you take a look at that on ultrasound, it could be spotty with some hyperechoic and hypoechoic um, locations, which means some look dark and some look light when you take a look at it on ultrasound. And the other thing that they do is, produce testosterone. So in regards to how it presents, so I mentioned the macroorchidism that you may see, and then also testosterone is produced. Testosterone has different effects when you compare it to estrogen, which is why precocious puberty looks different in boys than in girls. So when somebody is making too much testosterone, what you'll see is signs of virilism. So what that'll look like is you'll see maybe deepened voice, you'll see body hair, body odor, you'll also see some acceleration of the growth. But it's really the estrogen that causes your bones to grow the most and also close, but your body's making testosterone and not estrogen. So you may not see as much um, growth and um, and adult height growth truncation. With the caveat being that these boys are making more testosterone, and your body does have some enzymes that'll turn that testosterone into estrogen. So that's why you'll see the growth spurt. So all to say that in boys, precocious puberty is not as notable. One, because not all of them have testicular involvement. It's a little bit more of a low level testosterone production, so it may not be as notable. And then what you will notice won't be as dramatic as what you see in girls, because, maybe you'll see a 7-year old who's a little bit on the taller side, got some body hair and a little bit of a deeper voice, but you won't see things like breast development or vaginal bleeding.
So, um, as you mentioned that boys sometimes get estrogen also.
Um, so in general, all men make some estrogen. in their body. It's not to the extent of girls, of course, but that estrogen is involved in bone growth and maturation and development.
Mhm. So we've seen different treatment strategies. Some reduce hormone production, others block hormone effects. How do you decide which approach to take?
That's a good question. So, as I mentioned, when there's peripheral precocious puberty, the way it works in the ovary is it's a two-cell kind of thing. The granulosa cell and the follicular cell. One of the cells makes testosterone, and the other cell um, has a enzyme called aromatase, and it turns those androgens like testosterone and androstenedione into estrogen. The estrogen then goes and works on different cells like the breast tissue, the bone, the uterus. So, in regards to where you can have treatment, one of the medicines that people have developed is something called aromatase inhibitors. So, it prevents that conversion of those androgens to estrogen. And that's the thought, you stop that, then you decrease the production of estrogen. The other place you can also work on developing a treatment is okay, so the cells are making too much estrogen. Let's just block selective cells from the estrogen effect. So those are your two places you can look at. So you have the aromatase inhibitors that are a little bit more upstream and are supposed to limit the amount of estrogen production. And then you have medicines like tamoxifen that work at the actual cell site and prevent estrogen action. In regards to what people use, um, it's really kind of up to the clinician in regards to what they feel they're most comfortable using and things of that nature. So some people, they'll start, for example, with aromatase inhibitors if the aromatase inhibitors may work, it may not work, it may not be adequate, they could still have breakthrough bleeding, in which case they may either switch or add on tamoxifen, for an example, is one way that some clinicians may choose to manage it.
So what are the biggest limitations of current treatments?
Some of the big limitations, especially when it comes to McCune-Albright syndrome, um, is just how episodic the condition actually is in regards to the estrogen surges. There's currently no FDA approved drugs actually for peripheral precocious puberty in McCune-Albright syndrome, which is, an area of friction as you could imagine. So without that, the drugs that I mentioned are actually used off label. Um, Additionally, the drugs, they're not perfect and they're not always adequate for the patient. As I mentioned before, sometimes some people could still have breakthrough bleeding and they may have to switch drugs or use multiple drugs, which is an issue in and of itself. And that's just addressing one of the symptoms and effects of this estrogen exposure. So some may decrease vaginal bleeding, but you could still have some breakthrough bleeding. And then one of the other big effects is its effect on the bone. Remember the growth spurt and the bone maturation and the end adult height. Some of them it all depends on the person and how severe their condition is. But, some of them did show decrease in the growth rate but not always consistent improvement in bone maturation and what their end adult height could be. So all to say that it kind of depends on the patient, but especially given the sporadic nature of MAS, it doesn't completely always manage the vaginal bleeding, as well as the growth and bone maturation and development.
Mhm. So there have been studies using tamoxifen showing improvements in symptoms. Why did what did we learn from those earlier approaches?
Yes, yes. There has been a study in tamoxifen in girls with peripheral precocious puberty and MAS. And one of the things to also think about is one of the issues is with it being such a ultra rare disease, it's a difficult trial to run. But what it did show us is promise. Promise in that when tamoxifen was used, it did show some clinical improvement in regards to a decrease in vaginal bleeding as well as improvement in the growth. Um, However, there's some limitations, of course, in terms of how well the drug could work in terms of efficacy. But the big thing that it showed us is that tamoxifen is a good shows that there's a good mechanism of action in regards to the process, that this is a process that could work well in girls with peripheral precocious puberty and MAS.
So how does endoxifen build on or differ from tamoxifen?
That's an even better question because, as I said, that tamoxifen did show some clinical improvement, but there are potential places where it could be improved. So, tamoxifen is actually a pro-drug version of endoxifen. So what happens is when you give somebody tamoxifen, they ingest it, and then their liver breaks down tamoxifen into a number of different metabolites. I think there's somewhere around like 20 or so. One of the metabolites that it produces is endoxifen, and that is the hormone that works best in regards to what we want to see in regards to mechanism to actually helping it. The other metabolites that are made, could cause other side effects that we don't need. And then everybody's liver, depending on the enzymes that are there that breaks down tamoxifen, works a bit differently. So some people, they're more efficient at breaking down tamoxifen, while others are not. So you can imagine you take a certain dose of tamoxifen, your body's supposed to break it down, but maybe it doesn't break down all of it. Or maybe it does break it down and there's endoxifen, but then there's other hormones and enzymes that they have that may break down the helpful endoxifen that we want before it's able to do all the work that we want it to do. So with that, there's variable levels of the drug that we actually want in the body to actually go and do the work, which could affect the efficacy of tamoxifen. So as opposed to rolling the dice with uh, the person's liver and how well it breaks it down, we're taking you take that out of the equation and you just take the drug, the metabolite that you need, endoxifen, to give that to a person so that it can then work at the end tissue and you would have better exposure and more consistent exposure of what you actually need to help you there.
Mhm. You mentioned it's a prodrug. What is a prodrug?
Oh, sorry. Prodrug basically means it's the pre-version of the drug.
No.
So prodrug in this example is tamoxifen, and then it's metabolized by the liver to make the actual drug, which is endoxifen,
Mm.
Z-endoxifen to be precise.
Yeah. So is the goal to block estrogen effects or to, more precisely, target the pathway involved?
A little bit of more of the former, in that with endoxifen, your ovaries, your follicles, those cells are still producing estrogen. And with Z-endoxifen, we are blocking the effects of estrogen at the tissues that are concerning. The difference with Z-endoxifen and tamoxifen is that Z-endoxifen, as I mentioned before, is more directly the drug that's needed, and it will hopefully, hypothesis-wise, provide better exposure with potentially better results.
So what does the Z stand for? Curious.
So, as I mentioned, you have tamoxifen. It's broken down by the liver into, around 20 different metabolites. And for everybody who's taken chemistry before, all those different metabolites are somewhat different depending on what the enzyme actually did. So the Z doesn't necessarily stand for something. It's actually an isoform. So it's like, pretend you have a structure of endoxifen, and maybe there's a hydroxyl group here in the L formation. And then you move that hydroxyl group and you put it on this side. Then it's the Z formation, just to kind of give an example without getting into the chemistry. So all to say that there's similar versions. You can kind of think of doppelgangers, or for those who are into Marvel, variants of endoxifen. So one is the good one, the Z endoxifen that's going out there and stopping the inappropriate estrogen results. And then there's different variants that look similar to it, and they have maybe a different letter or a different number associated with them that don't quite do what Z endoxifen does.
The McCune-Albright syndrome can also affect other hormones, including in cases like acromegaly. Is there a common mechanism connecting these different endocrine issues?
Yes, there is. Again, when with McCune-Albright syndrome, the mechanism, you can kind of think of it as all gas and no brake. So particularly for the other endocrinopathies that you mentioned, like there's growth hormone, there's also thyroid hormone. Growth hormone is produced by certain cells in the brain. And generally, there's a higher level in your brain that says, hey, we need to grow you, somatotrophs, which produce the growth hormone. You need to make more growth hormone. In that case, your higher level of brain is not telling the lower part of your brain to make the growth hormone. It's just making it by itself. The gas is on, it's just making growth hormone. Um, and then you just have lots of extra growth. So it's the similar pathway in that it's all gas and no brakes, but they're in different cells. So the end result is kind of different. One produces too much growth hormone, one produces too much estrogen, one produces too much thyroid hormone, etc., etc., etc.
So with so much variability between patients, how do you design a clinical trial for a condition like this?
That's a good question, and that's a big part of my job to navigate those potentially difficult waters. So, with it being a rare condition, what we try to do is we try to do the most with the patient population that we have, and we design trials that will best let us know the efficacy endpoint if they're uh, if they're efficacious or not. So what we would do for something like this is we would, with the presentation being relatively so heterogeneous, we try to get a group of participants that are as similar as possible. So specifically for Z endoxifen, we would want to get a group of girls with McCune-Albright syndrome who are experiencing the peripheral precocious puberty to a severity level that is severe enough that we can actually measure a change. And then with those girls, we would figure out what their baseline um, severity is in terms of the burden. So we would see, for example, how frequently are you bleeding over a course of time? In this group of people? How much are you growing in a course of time in this group of people? And then once we figure out what their baseline is, we would then offer them the drug, and then we would see how that would change after time. And then from there, see if there's a significant difference. So in that case, each person kind of acts as their own control in regards to what the baseline is and to see where the change can be made.
So what studies are small? When studies are small, how do you determine whether a treatment is truly effective?
Sometimes that can be an issue. And in studies and science and research, we call that the power, the power of the study. How many participants are in the study? How long are they in the drug? So we can kind of adjust those variables to maximize the most amount of power. So with that being said, what we'll do sometimes to kind of adjust the variables if we have a small group of people, maybe we'll put them, maybe that will require a little bit more time on the drug. And then the other way that we can do that is the method that I described just recently in terms of having each person be their own control. So if a person comes in and they say, hey, we have bleeding episodes 5 times in the course of 3 months, then we'll see, okay, in the next couple of months, let's see how much we can decrease that number by. Hopefully we can get it down to zero. So from there we can say, okay, that's clinically significant,
Yeah.
as you can tell. I'm sure that would make a world of a difference in regards to the quality of life of the patient and their parents and caregivers.
Mhm. So what should patients know about accessing or participating in clinical trials?
I think what some of the important things they should know include that clinical trials are available, what to expect from the clinical trials. And, sometimes you can think of it as a source of hope in terms of that people are out there wanting to help them. So one thing that the first thing I mentioned that clinical trials are available, a lot of times when you have a rare condition, as you say on your podcast, 95% of rare diseases don't have any FDA approved drugs.
Right.
Um, so there are a group of people out there who are trying to change that. And it does take a long time in terms of expectations to run clinical trials. We want to make sure that the drug is safe and efficacious. So, first we have to develop what is a potential concept that we think will work, and then we try them in animal models to make sure that they're safe and that they seem efficacious, and then we then try them in patients that actually have the condition. So that could take a number of years. Um, so if you're waiting on treatment, clinical trials is one potential way that you can potentially, hey, get access to something that can help you something sooner than later. It's also a good way to potentially help out your patient community. If you're thinking about the altruistic side, like, hey, I'm helping and I'm contributing to that area of science. Next expectations, the researchers are doing what they can to provide solutions, and hopefully they work. So it's important to consider what are the expectations you have for the trial. Mm, reasonable, and hopefully they help. And that's what we're all hoping for.
Mhm.
And then hope, you know, generally there's a good mechanism of action and it's safe. So if anything, we're what we're doing is we're shedding some light, some rays of hope. This is me holding a flashlight of hope to provide solutions and treatments and options to those that need it.
Mhm.
So keep an eye out for those clinical trials. Sometimes your patient advocacy groups will let you know about opportunities. Sometimes you can search online. And if you feel so moved, feel free to participate and potentially help to make a difference in your community.
So the website to go to is clinicaltrials.gov. Uh, I know that not all of these drugs make it to market. Also, with the FDA, some of them get turned down, or lack funding? What percentage of the drugs actually make it?
Hmm. That's a good question. I forget the numbers exactly right now, but it all kind of depends on what stage and phase the clinical trial actually is in. So the further along they are in their trial the phase, the higher likelihood they are of making it. It's kind of similar to businesses. Sometimes, a
Yeah.
good number of businesses fail in the first 5 years. So generally how it works is, you have the in vitro models where you're trying it in like a petri dish, and you have the animal models. Then you try it in a small group of people who may not have the condition. That's called phase one. And then you try it in a small group of people that do have the condition. That's phase two to see if it's, safe and efficacious. Primary reason for phase one is mostly for safety. Primary reason for phase two is a mixture of safety and efficacy. Does this drug actually work? And then phase three is you try it in a big group of people that do have the condition. And that's again more safety and efficacy. So the further along you are in that process, the higher chance of success you have.
So what would meaningful progress look like for patients with McCune-Albright syndrome in the next few years?
So when thinking about that, it's important to think what would matter the most clinically for the patients and their providers. So McCune-Albright syndrome, particularly for Z-endoxifen, that would be the vaginal bleeding, and that would be the growth bone acceleration. So what would significant results look like? It would be a significant decrease or potentially cessation in vaginal bleeding while on treatment. And then the other part of it is the growth. We would see a normalization of their growth rate. How fast are they growing? And then also we would see that their bone age is keeping up with the actual chronologic age. So what that means actually is, if you think about it, estrogen matures your bones too much. So maybe you're a 5-year-old who has very mature bones and your bones are now the age of a 7-year-old. So we would want to see that slow down so that when you're 6 years old, maybe you have the bones of someone who's seven. And then when you're seven, maybe you have the bones of someone who's seven or seven and a half. So we would want to see the bone age come closer to the patient's actual chronologic age. Those are the big things we think the things that would make the biggest difference for patients and their families.
Mhm. So what gives you the most hope right now?
What was that?
What gives you the most hope right now?
Um, my whole career has been in rare disease. When I became a physician, the the motivation was to help people and as many people as possible. So when you're a physician, that group is your community of patients. And when you do research, that community then grows into everybody who's positively affected by the the breakthroughs that you make. So, I wanted to be as impactful as possible and really help patient populations and groups that had maybe the least amount of hope, that didn't have a lot of options. So the things that give me hope right now is that we're addressing and helping a community that has unmet need. And the drug that we have, we've already used Z-endoxifen for other indications. So the safety profile looks really nice. So that's very hopeful. And then, as you mentioned before, there was a tamoxifen trial, which showed that there are clinical improvements when you're on tamoxifen. And, the thought is this is uh, a more direct version. We have a more direct access, more direct delivery of the part of tamoxifen that we know already works. We're giving it to people. So I think with that, that's very hopeful that the mechanism of action, the drug will work relatively well, seeing how it worked in in previous studies, and then also seeing how good the safety profile looks like.
Mhm.
It was
So,
very reassuring.
so what interests you in rare disease?
Kind of just what I said. I really wanted to help people that need it the most. I'm a pediatric endocrinologist, so a lot of the conditions that we help manage are relatively rare. Um,
Mhm.
so that gives me a lot of exposure to seeing what these groups actually need and who needs the groups. And then particularly Atossa, where I work, it was a mixture of a lot of my loves, endocrinology, and then the other part, rare disease. So, particularly to most directly answer your question, I want to be as impactful as possible. Want to be able to grow old and say, I made a difference in the world. Uh, seeing patients, especially, the team, we recently went to a meeting for people who have McCune-Albright syndrome and hearing the stories that they shared about the burdens that they went through, I think was really motivating. We were already pretty motivated, but that definitely took us up another level. Even though it's a relatively small group, I mean, it's ultra rare, that doesn't make them any less of a priority in terms of developing strategies and treatments that they need. So, making a difference in the faces of the patients and seeing how much that affects their lives and their kids' lives and the caregivers, all it's a whole community that we're affecting, not even just the patients, it's the whole community that we're helping. So I like to do rare disease because I really want to make an impact in groups that I feel need more support and may have been relatively overlooked just because it's kind of difficult to run those types of trials. But giving hope to those who have less than others is something that I'm really passionate about, and Atossa is as well.
Mhm. So, do you have a connection to rare disease?
Do I have a connection? Um, I would say, me personally, I don't think I have a rare disease, except that being me it's
Yeah.
a rarity.
But I
Um,
mean, some people have children also that,
yeah. Without getting into all the personal stuff,
yeah.
I have as a pediatrician, as a pediatric endocrinologist, a lot of people come to me off hours and say, "Hey, Dr. Adebola, my kid needs help with this and that," and so I've helped friends and family who who had concerns about their children, and some of them actually I helped a while back. One of them actually had concerns for McCune-Albright syndrome. Their child was born with some big birthmarks on their back, and then they had some early breast development. So I gave them some advice, and they got worked up for McCune-Albright syndrome. Um, and then outside of that, with the other spaces that I've been in regards to helping different, I mentioned my whole career has been in rare disease. So, getting to know the patient advocacy groups and the big advocates, now that they're my personal friends as
Yeah.
well. So stay close with them. And then, my clinical practice, I was working at Hopkins before I kind of switched to this. Um, Even though they're not technically my blood, I still think of them as family, all one big community. So thinking about those patients and the impact that I was able to make in managing their chronic condition or diagnosing it, I think of that as that's my big connection. That's my family. My patients are my family, and helping them and seeing them progress from sometimes little small kids to then teenagers. Think about how much they've grown and just seeing how much progress they make and how much happier they are. That's my connection,
Mhm.
my family of patients.
Yeah. I've actually mentioned this before, but 30 million people live with a rare condition. That's actually 1 in 10.
Hmm.
So,
Yeah, if you ask some of my family, they probably think I have a condition too.
yeah,
How we're
there's over 10,000.
undiagnosed.
Yeah, there's over 10,000 rare conditions, so it's
Yeah, we all
a
have
lot
our
bigger.
quirks.
Yeah.
Hmm.
So when a condition affects different symptoms like bones, skin, and hormones, how does but comes from the same mutation? Does that change how we think about treatment, the disease is treatment is of the disease as a whole?
Yes, it does change things a bit. You know, sometimes when people have a rare condition, you have one source of where the pathology originates. You know, maybe there's an enzyme that doesn't work well, and then you overproduce a substrate, or you overproduce one of the end products because one enzyme doesn't work. And then that's what causes the issue. So sometimes with those conditions, you can just target where that enzyme issue is, where that substrate or product issue is. However, with McCune-Albright syndrome, um, it's different in that we can't just target, at least at this point, at this time, where exactly the pathology happens on a cellular level. We can't necessarily just stop every place that has GNAS and say, "Hey, you got your gas on, take your foot off." You know, we can't do that. So we have to, and we can't do that because it affects different tissues. It's not uniform in regards to what tissues are affected. So every person's kind of different. Some people may have primarily bone issues. Some people may have primarily thyroid issues. Some may have mostly estrogen issues. Some people may have a mixture of all of them or two out of the three. So when it comes to treatment, as opposed to other things, you can't just necessarily treat one upstream location. You'll have to treat the person individually in regards to what where the burden is. Um, so you'll have to treat the bone to a certain amount and say, hey, you know, we can use this to potentially help with the bone. There's another portion, if there's issues with pain, we'll have to treat the pain. If there's issues with estrogen, we'll have to try and prevent that estrogen action. So some patients may just need those two, while some may need, oh, well, you know, I also have thyroid issues, so you have to treat it. So in a case of MAS, it's a little bit more à la carte, as opposed to some other conditions where you can just kind of treat the upstream pathology.
Mhm. So thank you so much for listening to this episode of Our Connection, and a big thank you to Dr. Adebola Giwa um, for helping us better understand the many different phases of McCune-Albright syndrome. Um, One of the biggest takeaways from today's conversation is that that no two rare disease patients are exactly alike. Even with the same condition, symptoms, complications, and treatment approaches can vary dramatically from person to person. McCune-Albright syndrome, the variability is built into the condition itself through mosaicism, affecting different systems in different ways, including the skin, bones, hormones, and development. We also explored how conditions like precocious puberty are being approached through different treatments strategies, including therapies like Tamoxifen and newer approaches involved like the Endoxifen, while also touching on the broader challenges of clinical trials in ultra-rare disease communities. Through finding enough participants can be difficult. If you found this episode helpful, make sure to follow and subscribe to Reconnection on your favorite podcast platform. And if you're watching on YouTube, don't forget to subscribe to my channel, Rare Chef, and leave a comment below. And share the episode to help raise awareness for rare diseases and the families navigating them. Now, for audio listeners, you can always send me a text link in the show notes and share your thoughts and connect directly. You can also follow our connection on Facebook, X, and LinkedIn for more episodes, updates, and advocacy conversations. And as always, this podcast is for educational and awareness purposes only. Should not replace medical advice from a qualified healthcare professional. If you or someone you know is struggling emotionally or in crisis, help is available. In the U.S., you can you can call or text 988 for the Suicide and Crisis Lifeline, 24 hours a day, 7 days a week. Until next time, keep advocating, keep learning, and keep connecting.